Hello students, I am Sonali. Welcome to my class of biology. Today we are going to learn about artificial hybridization technique and double fertilization technique. We will see first what is meant by artificial hybridization. So it is one of the major approach used in the crop improvement for better yield and for better crop production, more crop production. So only the desired pollen grains, which pollen grain desired, not undesired. Pollen grains are hand pollinated. Main word is hand pollinated and are used for the fertilization. And in artificial hybridization, two techniques are used, that is emasculation and bagging procedure. So what is this emasculation technique? It is the technique of removing the stamen or killing pollens of a flower by cutting the anther without the female reproductive organ. This is known as emasculation technique, and this prevents the self-pollination. Now, what is this bagging procedure? This bagging procedure used to prevent the fertilization of a stigma from undesired pollen by covering the emasculated flower with butter paper or polythene bag. See in this picture, this emasculated flower is covered by butter paper or polythene bag. Now, because only this is used for in the plant breeding program also, because only the desired pollen grains are used for pollination and protection of the stigma from contamination of undesired pollen grains, right? So these two techniques are useful for artificial hybridization. Now, what is meant by double fertilization? It is a complex fertilization mechanism in flowering or angiospermic plant. It was discovered by Navashin in 1898 in the Lilium or Fritillaria plants. Now, what happened in double fertilization? Just fertilization means fusion of male gamete with the female gamete, that is known as fertilization to form a zygote. Right, in double fertilization, one male gamete with the egg, fusion of one male gamete with egg, that of another male gamete with secondary nucleus, this is known as double fertilization. We will see how it is occur. So, for the artificial hybridization and for the double fertilization, the main important point is entry of pollen tube into the ovule is necessary. So, the porogamy, mesogamy and chalysogamy. This one porogamy, mesogamy and chalysogamy. These three techniques are, these three phases are important. Right. So, see, this is the chalysal end. These are antipodal. This is integument. This is micropylar end. In porogamy, what happened? The entry of pollen tube from the micropylar end into the ovule. Right. So, this is known as porogamy. Now, second one is mesogamy, means entry of pollen tube through integuments into the ovule. This is known as mesogamy. And third one is chalysogamy, the entry of pollen tube through the chalysal end. These three phases are important. Now, what happened in double fertilization? First point is syngamy. In syngamy, it is the fusion of haploid male gamete with the haploid female gamete to produce diploid zygote. Whereas in triple fusion, second haploid male gamete fuses with diploid secondary nucleus, producing PEN, means primary endosperm nucleus, that develop into triploid endosperm. Zygote develop into an embryo. Syngamy is a type of generative fertilization, whereas triple fusion is a type of vegetative fertilization. Now, here both the male gamete participate and therefore it is known as double fertilization. Both the male gamete are participating in this technique, in this process, so it is a double fertilization. Now, all these points I explain you from the diagram. See, double fertilization process. What happened here? Stigma, pollen grains. Now, here ovary part and this is ovule. It is a female reproductive part. Now the pollen tube is here, which carry the two male gametes. Now here in this diagram, when this male gamete enter into the ovule, what happened? In this diagram, we can invert it also. This is the micropylar end and this is the egg. So the two male gamete, non-motile male gamete are present in this pollen tube. Right. Now, one male gamete is fused with this egg and another male gamete, means sperm nuclei, is fused with this polar nuclei. That is secondary nucleus, and this is known as primary endosperm nucleus, or triple fusion is occur here in the embryo sac. So this is the double fertilization technique. This is thin gamete and this is the triple fusion technique. Now, significance of double fertilization. So, what is the use of double fertilization? It is a unique feature of angiospermic plants, means flowering plant. It ensures that parent plant invests seed with the food store only if the egg is fertilized. That time, egg must be fertilized. The diploid zygote develop into embryo, which consequently develop into a new plant. Triploid primary endosperm nucleus develop into nutritive endosperm tissue. It can provide a nutrition to the embryo, so it is a nutritive endosperm tissue. It restores diploid condition by fusion of haploid male gamete with haploid female gamete, that is through syngamy. Now, it also helps to avoid polyembryony. Polyembryony is nothing but formation of many embryo rather than one embryo. It is known as polyembryony, that is from single fertilized ovum or from single seed. So, it can be avoided in the double fertilization technique. Now, what is this post-fertilization changes? In post-fertilization changes, development of the endosperm. In post-fertilization changes, development of endosperm is formed. That is, first point is triploid primary endosperm nucleus, that is PEN. Repeatedly divide mitotically to form a nutritive tissue, and that is known as endosperm. Means this endosperm is provide nutrition to the embryo for the growth. In post-fertilization changes within the ovule, the embryo and endosperm are seen to develop simultaneously. Means in the ovule, this embryo and endosperm is developed, and this endosperm is provide nutrition. There are three types of endosperm on the basis of mode of development. So, which are these three types of endosperm? That is nuclear type, cellular type, and halobial type. See the diagram first. Nuclear type. This free nuclear, here it happened at the micropylar end, and see the nucleus at the edges, at the periphery. Now, in the cellular diagram, what happened in cellular phase? It divides and it forms the cell. It can divide into the cell. Now, halobial type means it happens in chalice. This is chaliceal lens. It divides into two parts. Major part of the micropylar end and minor part is known as chalice. Thank you, students. Thank you very much.